Basically what we're doing is we're taking the One Health paradigm and putting a time frame on it, extending it into the past. And that's really important because if you study history, you need to study history to understand why the world is like it is today. But if you want to understand the disease and health landscapes that we have today, you have to look back in the past.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I'm Kim Klinowski, your host for today's episode. Before modern medicine, disease still left a signature, and today's guest has built a career learning how to read it.
Our guest today is Dr. Elizabeth Uhl, faculty member in the Department of Pathology at the College of Veterinary Medicine. Betsy, welcome to the podcast.
Thank you for having me.
So, Betsy, our first question that we like to ask our guests is about their pathway to science. So what got you interested in science either initially or really kind of set that flame afire?
Well, both my parents were botanists. That was one area I was not going into. My father also had a great love of history, so the interest of both of them in science was rather broad. I have 4 siblings. 2 of them went into historical careers and 2 went into medicine. My sister's an MD and I'm a DVM, so we kind of split the interests of my parents.
I love that. So they clearly had a very strong influence on you guys growing up.
They did, yes.
Did they marry the 2 at the time, or was it really just kind of 2 separate interests that you married?
Well, my father used to take us on plant collecting trips all over Canada, U.S., and Mexico. He studied succulents, not cacti, but the little sedums and things. My mother studied palms, so they had interesting debates over which was easier to collect. But there was just a passion for both, and the historical side came from my father. He had grown up in Atlanta. He heard Civil War veterans talking about the war. He found mini balls in the backyard, and it just set up this real interest in it. But he also had really broad interests across the board. So I think what's really important is somebody's passion can be infectious, and so that was part of it.
Mm-hmm.
Well, this is the place for infectious.
Oh, that's for sure.
Betsy, you're actually the first pathologist we've had on the podcast, so I think the next question is, what's a quick definition of pathology?
Well, pathology, the actual meaning of the word is the study of suffering.
I did not realize that.
So it's like pathos.
Yes.
And so that's an interesting one. What we do is we study disease, and when I used to go around and ask people why they went into pathology, and a fairly common answer was that the disease was more interesting than the cure. So it takes a certain, I want to know all about it. You can often treat effectively without knowing all about it. So I think it's a certain sort of personality for that.
And what are the day-to-day duties of a pathologist?
For veterinary pathology, there are basically 3 hats we have to use. One is diagnostics. So that would be necropsy. We'd go down to do autopsies on animals. It's one of the things. We also read biopsies where people have taken a piece of tissue out and they want to know what the tumor is or whatever. Mm-hmm. So that's the diagnostic service part. There's also research, which is investigating disease and looking into it. And then the final one is teaching. Mm-hmm. So sometimes we joke that our assignment is 50-50-50.
I know how busy you guys are when you're on rotation and you have all your path duties to do. Are there specialty areas in veterinary pathology?
There are, and that generally follows people's interests. So my area of diagnostic interest is bone pathology. I've always enjoyed that. The nice thing at UGA is we have enough pathologists that people could have time to specialize. So we have people that are interested in eye pathology or skin pathology or neuropath or reproductive. When we're on service, we have to cover everything. So So there's a baseline from which you can expand.
You might specialize, but you also have to be a generalist.
Yes.
And do all veterinary students have to go through a pathology rotation or learn pathology? Is that I just don't know a lot about the veterinary curriculum.
Yes, we have some teaching in the veterinary curriculum. The students will rotate through necropsy.
Okay.
Not the biopsy side, but the necropsy side.
So we know that you've got several research areas. One in particular, I think, may be new to our listeners, and that's paleopathology. Can you explain that to our listeners?
Paleopathology is looking at disease in animal remains, usually from archaeological sites or historical sites. So it was sort of a natural expansion from my interest in bone pathology, since most of the time you're dealing with skeletonized remains, although the Egyptians mummified a lot of animals, and people are starting to look at the pathology in them. It's an interesting specialty in a way. In human paleopathology, it's really a branch of archaeology and anthropology, so it's not coming out of medicine or path in general. And that's an interesting way that things get set up. Veterinary paleopath or animal paleopath is relatively new. The disease in animals just in the past has not been investigated like it has in people.
So has this interest been, we talked about your love of history and growing up with history as well as science. So was this something you actively kind of went into, or was this a kind of a parallel interest that you had? I guess the question I'm really asking is, how did you get interested?
How did I get into
Yeah.
paleopath? Well, it turns out that UGA has a pretty good zooarchaeological collection, and there's a zooarchaeologist here, I think she's recently retired, also named Betsy Wright. And I saw that she was here, and I just contacted her saying I was a veterinary pathologist, and did you see lesions in the bones? I'd be interested. And she was very interested because, she can tell that it's the left distal toe bone of a turkey. I'm not as good as identifying bones, but I can say, yeah, that's not normal. So it was a sort of natural complementary of expertise. And then she connected me with an archaeologist at Georgia College and State University. Unfortunately, he's passed away, but he had excavated over 100 pre-Columbian dog skeletons out of Virginia. So those were the first ones that I got to look at. Then I realized when I'm on the necropsy floor, I don't look at lesions and dried bones. That's not how we look at disease. So I had no idea what even the common things look like in a skeletal remains. So that's where I had to start learning. And there aren't really good collections or reference collections for doing that, unlike in people. So I've been working on an atlas to try to say, okay, I know that this was this disease in this animal bone, and this is what it looks like in the dried bone.
Wow.
I guess 500 or 600 years could cause some changes in a bone.
Yes. Yeah, and, holes in bones, they're not always very specific. So that's where you have to say, what do we know about this animal? What diseases are common and which ones might show up in the skeleton in this way? So that's where having an understanding of disease in animals is really helps what we call narrow the differential. You may not know definitively what it is, but you've got a list of what most likely it is.
What can you learn from studying paleopathology, or I guess more specifically the bones, but I'll let you decide if we're talking generally or specifically.
Well, one of the challenges, especially in animal paleopathology, is that we didn't bury whole animals all over like they did with people. So a lot of times you're dealing with really fragmented, scattered remains. And a lot of times you don't. And the other thing is that when the horses in the cities died, they were processed completely for all kinds of products. So some diseases we just don't have, shall we say, hard or evidence, to excuse the pun, for them. And there's also a paradox, which is that a lot of diseases don't leave lesions in bones. So while it's good to try to identify the lesions we do have, I also joined with Jane Bykestra, who's an absolutely amazing woman. She's at Arizona State University. She is a member of the National Academy of Sciences. She started bioarchaeology because she wanted to get science into archaeology. And I met her at the Paleopath meeting, and we've been working together to get back to your question, we've taken what we call a one paleopathology approach. And that means that we're doing more than simply looking at holes in bones. Okay, so we're trying to go back, look at historical information, ancient DNA, what we know about what societies were like, how, in this case, how animals were handled. A lot of historical information is really needed to understand animal diseases. So that's really been exciting because you can really expand off of, well, here's a hole in the bone and it could be one of 5 diseases and we don't know which one.
Sounds like a really holistic approach. You need multiple points of view, multiple sources of evidence.
Yes, it's very cross-disciplinary, which is also quite exciting. Basically what we're doing is we're taking the One Health paradigm and putting a time frame on it, extending it into the past. And that's really important because if you study history, you need to study history to understand why the world is like it is today, why we have the countries we do, why we have the conflicts we do. All of that's coming out of understanding history. But if you want to understand the disease and health landscapes that we have today, you have to look back in the past to understand where they came from. It's very interesting. Yeah, there are challenges to it, but you also get to find out about some interesting other specialties. For example, there's a whole specialty called medical geology, which I hadn't known anything about.
Oh, I've never heard of that.
But that's, there are textbooks on it, and they're looking at the geology of an area and what minerals or other things were there that might have led to problems. And one interesting example that comes out from taking that approach is China, for example. Horses were the cruise missiles of their day. They were very, very important for the military. And China didn't really set up a good horse breeding program, and it wasn't exactly clear why. Even when the Mongols came in, they weren't very successful in breeding these really top-notch horses, which came from different places. And it turned out that one possible reason was that the area where they were trying to do this was one of the most selenium-deficient areas known. And selenium, I know from upstate New York, we used to see foals coming in with selenium deficiency, white muscle disease. So that could have been a reason why they weren't very successful in trying to breed their own horses.
And is selenium deficiency something that you see evidence of in ancient bones?
No, really.
Okay, too bad.
Sometimes you can see some evidence of toxicity, which actually, that's the other side of it. When I was in college, my advisor was a nutritionist who had studied selenium and said that the area where the one reason that Reno didn't get to relieve Custer was because his horses and mules had selenium toxicity. They'd been kept on an area where selenium was really high, and they were literally losing their feet. So I'm not sure it would have made a difference in the outcome of the battle,
right.
But he did take some blame for not getting there. And if you look at the history of the description of what was going on with the horses and where they had been overwintered, it's quite likely that was an issue.
And that's another intersection of history and medicine. So
Exactly. So finding those connections is really, really interesting.
So, Betsy, are there some diseases today you think we misunderstood because we didn't consider their historical context?
Well, we haven't been very good at predicting the next pandemic. And I think that one reason for that is that we've tended, for example, if an animal gets a disease that can be passed to people, the main emphasis is on a zoonosis, that the animal gave it to people. I got interested in evolutionary medicine, and when you're looking at infectious diseases from an evolutionary standpoint, the most important thing is opportunities for transmission. And what you realize is that a lot of times the way animals were kept were setting up pathogens for transmission, and oftentimes pathogens they didn't get in the wild, or at least not to the same effect. So I think we've missed, because all the emphasis has been on how the animals gave it to us. And if you look a little deeper, a lot of times there were people that either gave and they've done some studies saying some of the viruses, there are more viruses passed from humans to animals than the other way around.
Oh, really?
Yeah. So you understand, why the interest is on the human side, but I think if you really want to understand the diseases better, you have to look broader than that. That's where I think a broader approach could help a lot. Because when you think about what people did, they brought animals into very crowded conditions. And, you look at how animals lived in the wild. Let's say horses, they were in small herds or smallish herds that moved around a lot. And all of a sudden, you've got, in New York City, there are 500 horses per square mile. So that to have them crowded and mixed, they didn't mix like that, that's going to set up a whole nother scenario for the pathogens that they get. And why? So some of the animals probably wouldn't have been a problem in the wild because they're in smaller groups that don't mix like that.
I have to say, today I was looking over a book chapter you read, and there was a figure there that I think relates to what you're mentioning right now, and that's the relationship between transmission dynamics, which you've just alluded to, and pathogen virulence.
Yes.
Explain what the interaction is there.
Well, if you view it from the pathogen, there was an old saying that it's not in the interest of a pathogen to kill its host. And it's like, well, that depends. Because from an evolutionary standpoint, the interest of the pathogen is to get to a new host. And if it can do that by killing the one it's in, and by multiplying a lot and killing the one it's in, that's can be a selection for more virulence. So tuberculosis is a good example. It's a disease that's been around for a very long time. And when you think about hunter-gatherer groups that are small, so it's either you have a pathogen in a small group of people or animals. And in that scenario, it may not be in the interest of the pathogen to kill a host because it takes longer to get to the new one. So if you look back into TB, some of the earlier strains of it weren't as virulent. Then in Europe, you had a lot of people crowded into cities, a lot of people coughing, and that could potentially have put pressure on the pathogen to become more virulent. There wasn't a cost to it. So It's a cost benefit, really. If a pathogen kills its host quickly, the cost is there's less time to get to a new host. The benefit is they're more the pathogen. In a sparse population, you have fewer, the benefit of not killing the host is you've got a longer period to get to the next one. That's one way to look at things, and it can explain some of the increase in virulence that we've seen in some diseases or some pathogens.
Can one paleopathology provide evidence of the effectiveness of a response to disease?
Oh, yes.
Or failures, I guess we should say, too.
Oh, yes, both. There's a disease in horses that was really, really devastating in wartime called glanders. It's Burkholderia, Malii, which it's been a really bad pathogen across time. And it's contagious. Usually horses kept in large groups. It's even been called the equine TB because it's bad. But you can have horses that kind of carry it for a bit. Apparently there was a period in France where they didn't believe it was infectious, which of course is going to change how you deal with horses that have it. And there were good reasons they didn't want to say it was infectious, because you might have a very valuable horse that's still functional, and you don't want to cull it or isolate it or not use it. And that was about the time that Napoleon went into Russia. And the interesting thing about that is that, and he knew this, he could replace the men he lost in Russia, but he could not replace the horses. And so, yes, it was pretty horrific, and a lot of them died of the conditions, but another problem was glanders in the French horses.
That's another fascinating historical connection. Kind of a brutal calculation he had done there.
Well, he was told to make, he sent in an incredible 600,000. It was some incredible number, huge. And then he was encouraged to start another cavalry unit. He said, I don't have the horses to replace the ones I've lost already. And that's something that's sometimes forgotten in the history, too, is how much the animals impacted and their diseases, which is an area that I'm interested in, how the animals and their diseases impacted our history. And I think there's a risk of the animals becoming history's ghosts because we don't live close to them as much as we used to, so people aren't as aware of what their influence was.
If you get in skeletal remains of an animal and you're asked to figure out if there's clear evidence from the bones of what it died of, maybe rank them. What are the easy diseases to find and what are the tough ones to figure out as a pathologist?
One of the things we can tell from the skeletonized remains that we don't look at so closely in modern necropsies is that we can tell based on, let's say, evidence of degenerative joint disease and other things, the forces that were impacting that animal, and potentially get an idea, let's say, how they were moving, how they were working, so we can tell a bit about their life. Again, it's really hard to, let's say, take a horse and get it down to a skeleton. We don't usually do that. So we don't usually look at the bones that closely in modern necropsies. But I was involved with a study with a colleague in the United Kingdom where they'd found this collection of horse bones and it was just knackered, just little, they were just scattered. You couldn't tell, there were no complete skeletons. He noticed that there was a raised area on the bone of the radiuses of several of these horses. And we identified it as the attachment site for the check ligament for the superficial digital flexor tendon. And as we looked into it, it turns out that horses, when they're running at a gallop, they hyperextend the knee, and that puts stress on that tendon. It's a problem in modern racehorses, which also would have put stress on the check ligament, and the body's response to that would have been to reinforce that attachment site with more bone. Turned out that the site that he'd been digging, there had been a racecourse on that site. So from a single bone, and he had several of them that had this, and then we made comparisons from anatomy collections mostly to say how prominent is this normally. And from our investigation, it looked like it was something that would have been enhanced by how the horse was moving. And the other thing they did was, there was a study in China where they had horses that had been buried, sacrificed. So they were in place, and you had chariot horses and you had riding horses, and they were actually buried in situ with the chariots and other things. And they looked at where they saw bone lesions. And most of them were in the thoracolumbar spine, which is someplace we basically never look in necropsy. So I'm going, we ought to be looking at that a little more, I think.
Interesting.
And that's where the paleopath can inform modern investigations.
Right, and maybe thinking about how you treat animals that are working under certain conditions, like understanding what it's doing to their body, I imagine.
Yeah, well, there's another study we worked on with the archaeologist for navicular disease. It's a disease there's a small bone in a horse's heel called a navicular bone because it looks like a boat, and it's basically sort of a pulley for the attachment site of the deep digital flexor tendon, and it is a common site of lameness in modern horses. But looking at the history of it, it wasn't described until, this is where the historical references come in, especially the historical veterinary ones. So there's a veterinarian saying, we didn't see this until 1840. And sometime between 1815 and 1840, it got common. And I was thinking, well, maybe that's when they paved the roads, because you have more concussion on the heels. But a lot of interest was put into horse medicine because horses were the cruise missiles. And that if you go back through history, you don't see that disease described. And then I asked the archaeologist, and again, this is the problem, we only have scattered navicular bones, but we had some from medieval, post-medieval, Neolithic, and we just didn't see it. And they had also identified in the 1840s that it was a disease caused by work. So you sit back and go, okay, maybe we need to look at how the horses are working if we want to understand, and what changed. And then the interesting part of that, too, when you start looking for evidence in the past, you even use art. So we looked at, you've got 30,000 years of depictions of horses, and none of them are showing horses moving heavy on their forehand, which is what would have caused navicular. And so the art can be really informative, and that brings in, again, the multidisciplinary part is.
Yeah, I never would have thought the art, but that makes sense now that we're talking about it.
I love that. That is so cool.
We had another one we've been looking in, which is typhus. And typhus is a terrible disease. It was shipboard fever, gall fever. It was a problem in a lot of wars. One of the interests that I have is how diseases got to cross continents. The other thing about typhus is it can infect other animals, but they don't pass it on, with one exception, and that's the southern flying squirrel. And when I first saw that, I was like, how did that happen? It's a human European disease that's carried in southern flying squirrels in the U.S. And it's still a puzzle, but again, art and history can give a partial answer, which is that squirrels were really popular pets in the early 1700s when we know typhus was coming through. And there's even a painting showing a little boy with a flying squirrel on a leash. So there's a possible way that it got in. And a similar tie had been recently made a few years ago in a great study out of England where they know that red squirrels in the UK can carry leprosy, as do armadillos in the US. And when they sequenced it, it matches a medieval human strain. So you're going, how did that happen? They excavated bones from a leprosarium in a site in England, and they got a strain of leprosy out of it. And not too far away was a furrier with squirrel bones, and they got leprosy out of the squirrel bones. Turns out that squirrels were common pets in the Middle Ages. A nice painting of a medieval lady with a squirrel on her lap. And they also were used a lot in furs. So they had the alternating sort of triangular shape, light, dark. Those were squirrels' front and backs.
So the more contact, the more potential for transfer one way or the other.
Right. And it's unlikely the squirrels gave it to the people.
We'll clear the squirrels of this one.
Yeah, we'll give them a pass, I guess.
So one thing you mentioned when we chatted before was that measles might have jumped, and I'll let you clue us in as exactly how solid the evidence is, jumped from humans to dogs and is now a disease that we know of as canine distemper virus. So tell us that story.
Okay. Well, it started with the pre-Columbian dog bones that I was looking at because I'm going, okay, what kind of diseases could I expect to see? And we know that canine distemper, if it infects puppies, causes enamel erosions in the teeth, and the dogs will have them. So I said, okay, there's one. Also, historically, the Native American dogs were relatively quickly replaced by European dogs. So I thought maybe it was a disease. But then I started looking into the history of canine distemper, and a very different story came out. Canine distemper was first described in Peru in 1740. The evidence was pretty strong. Europeans, and then it spread to Europe in the 1760s, causing devastating outbreaks. It wiped out the French royal kennels. There were talks of hundreds of dogs dying in Madrid. It was the first disease where they had a crowd-forced vaccine. So, and it's not a disease that could be there and you don't notice. I mean, the morbilliviruses are really devastating. The other thing is that measles, distemper, and rinderpest are some of the most closely related viruses known. Monoclonal antibodies will cross-react with them. You can take genes from one and put it in another, and they still function. So I looked at that and I said, there's no way that that virus could have arisen in the Americas and been isolated from measles and distemper, or measles and rinderpest. And rinderpest never really got here, small outbreaks, but measles did. And measles was really devastating to the Native Americans. They were so sick they didn't have anybody to take care of the sick, let alone bury the dead. We're talking, almost close to 100% morbidity and mortality. And in an area in South America, there were more people and there were also more dogs. So you have a lot of unburied bodies around. So it would have been an ideal situation for the virus to jump. The other thing is when you think about how dogs got into the New World, they came in with, most of the genetic studies are saying that they were domesticated in Europe or Eurasia and then came across with people. But they came across in small groups. And distemper, measles, rinderpest, you need large numbers because they're diseases that you're either dead or immune and nothing in between. That's why it became a puppy disease or a childhood disease. That's a large population of non-immune individuals.
Right.
So if you think about how dogs actually lived, it would be in small packs, it would be unlikely to have arisen that way. Now, what's happening today is you're getting a lot of canine distempers jumping species, and that's probably from, again, crowding. And again, you've got a virus that is jumping to other species. It's got opportunities for transmission. But the other thing about measles and canine distemper and rinderpest is they say they use the same two highly conserved receptors. So they are so close, and the evidence that we couldn't get a virus out of a dog mummy or something to check it. But if you put all of the pieces together, that's at least tells you potentially where to look. And then the other thing was with Jeff Hogan, I went to Jeff Hogan and I said, well, is there anything about the virus that we could tell whether it had been in humans? He suggested codon usage bias, which is, you've got 20 amino acids and 60 different codons. So you've got duplications for the codons for which codons and amino acids. And their species have preferences as to which codons they're using for which amino acids. So there's a bias. So we looked at it across the canine distemper genes, and all of the canine distemper genes had a codon usage bias that was closer to human than canine. Viruses they don't have tRNAs, so they have to match the codons. And that was, again, a very, it's a soft finding. But it had also been noted that codon usage in some pathogens were closer to, pathogens don't have to adapt exactly, but they would be closer to human usage than pathogens that were primarily in other species, which is also an interesting observation. Could simply be they infected more people. I don't know.
And I guess that also speaks to the fact that it hasn't been historically all that long since the virus jumped from humans to dogs. So it hasn't had the time to adapt to the canine codon bias.
Well, there probably isn't a whole lot of selection pressure on it to do that, because the mammalian codon usage bias is pretty close. So as long as it's working, and there could be some selection not to if it's jumping species. So it has to be close enough, but not
perfect.
Not perfect, because then it can only infect that host. So codon usage bias is an interesting, complicated but interesting. The other thing I think that hasn't always been considered is they'll say, okay, time to common ancestor on some of these viruses. So you look at canine distemper virus, and some studies say that was, 1800s in Kansas or something. And it's like, the problem with that is that all of the morbidity viruses, their RNA viruses, selection pressures are varying, and all of the information on sequencing is from viruses that have after vaccines have been exposed. So some of them have been under pretty high vaccine selection.
Yeah.
So trying to go back 500 years to what the virus was like based on what it looks like now we have to do that really carefully. Because all the sequences we have are recent.
So Betsy, I understand that you were just awarded a Special Collections Fellowship, which I think allows you, you're the perfect person for this, right? To go into archives here at UGA and to then develop something to use for teaching.
Yes, it's just started. So we just had our introductory session. Again, as I've started to look into this, I've realized how important the historical sources are. And the other thing that can be tricky is that especially if you've really studied diseases and you look at the descriptions, they all sound the same. So it's easy. Sometimes I'll be reading papers and they'll speculate on what disease it was, and it's like, I don't think so, based on my experience with it. So I'd like to bridge that gap a bit. And going back into the historical records, and then also potentially with the students, connecting with a lot of the information we have about animal diseases isn't published. It's in archives of cases. So in diagnostic labs and other things, because we only write up the zebras, basically. And that's a problem if you have people from outside the profession coming in, because they'll think that a disease that is in our literature is common, and that's probably what it is, and because we don't always write up the common stuff. But for the students, and I'm excited about it, because going back in the late 1800s, somebody wrote a book called Animal Plagues, George Fleming, and what he did was he went back 2,000 years to any mention of an animal disease and put it in his book. And it's frustrating because, something was killing cats in London in 16 whatever. That's all they say. But it's in people's letters. It's in descriptions and other things. And there was also a person at the USDA that did that for the US, which was also kind of interesting in the 1930s. It talked about bees collapsing. So there'll be these really interesting little tidbits that you're trying, that's often where a lot of the animal diseases were mentioned. And it's something, too, to set a project for students to do.
Is this for veterinary students, or do you want to do it for undergrads just in general?
The vet students, as we used to say, it's like trying to drink from a fire hose.
Yeah.
They don't have any time.
Yeah, I was just thinking but I just think the applicability for undergrads could be even greater about thinking about history in lots of context.
Again, I teach an evolutionary medicine course for undergrads for that reason. So I'd like to incorporate things into that because I'm doing a lot of history of disease in that. So potentially graduate students, there's some things that we have a student that's working with us now. It's sort of a side project. We've been looking at how pathogens crossed the ocean and what were the features that allowed some pathogens to become pandemic ones and others not. And you have to go into a lot of the history's importance. So, for example, Hawaii didn't have any mosquitoes before.
Or avian malaria.
So when did they get in? And we know, we found a record of a ship that came in there and dumped its ballast with mosquito larvae in it. And then a few pet birds with malaria, and there you go.
So I do want to put in a shout out to Special Collections because I take my first year Odyssey seminar class there for a couple class meetings, and the folks there are so helpful. And they've got, I think, around 6 million historical records. So it is an amazing resource. They're great people to work with.
And anybody can use it. You don't have to be connected to UGA.
Yeah.
They will, again, I've only had one session with them, but I'm really looking forward to it. And they were stressing that anybody can use the collections.
Betsy, when you're not tracking ancient disease outbreaks or thinking about bones, what are you doing in your free time?
Well, I'm addicted to horses, so which has a connection because I've been interested in the pathomechanics or basically whole body pathomechanics of degenerative joint disease in horses. And so it's tied in with my passion for riding. So my research and passion for horses have sort of merged. And I've also been really interested in how they can synchronize to us, because they do. They will synchronize to our movements. You can walk next to them and they're in perfect sync with you. So how do they do that? You got two different species.
It's fascinating that they do it, however it's done.
Yeah, well, I've been working with some engineers to look at that. There's a new concept of how the body works, which is that we're really tension-based. It's called tensegrity. So could that kind of structure facilitate synchronization, the structure itself, because it happens too fast for nervous signals to go through. And then when you think about horses, you've got a person on a horse performing as a single athlete at Olympic level. That doesn't happen by pull on the rein and give them a kick. So
that also doesn't happen when I'm the human on the horse. That's been proven multiple times.
Yeah, it takes a bit to sync. But I've been working with Jean-Luc Cornille at the Science of Motion. He's down in Eatonton. He's amazing because he was a French 3-day event rider and has been working in training and working with horses with lamenesses. And we're saying, okay, he'll note a certain body posture or way they're going that goes with the horses that have a lameness. And then when that's corrected, they can be sound. So, and I have a anatomist colleague who's currently at LSU, but at Michelle Osborne. She'd been my postdoc, and she's coming here, which is great. So we have a 3D dynamic horse skeleton that we can put in different positions and analyze the postures to say, okay, here's an image of a horse working. We can put the skeleton in that image, and then we can say, oh, look, here's why it's got compression in the neck or wherever. So we're hopeful that can be useful.
Very cool.
Cool, yeah.
Very cool. Well, our guest today has been Dr. Elizabeth Uhl, faculty member in the Department of Pathology at the College of Veterinary Medicine. Betsy, thanks for being with us.
Oh, it's been a real pleasure. Thank you for having me.
Links to additional information about Dr. Uhl's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today, and if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes exploring the research being conducted at UGA. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemeth, for research, editing, scheduling, and keeping us on track.